we will begin our discussion on protein metabolism, which is just another way of saying translation, protein translation. As a process, protein translation is perhaps the most complex biosynthetic pathway, which, as you can see from the slide, involves approximately 300 different biomolecules, and they all synthesize new proteins with a rate of 20 amino acids per second. I want you just to take a pause, perhaps even pause the video if you like, and think about how complicated this must be. How all of these molecules need to work in coherence and cohesiveness and together to try to accomplish this humongous task. And then think about the regulation, and to add to it, think about that this is happening in the cell while all the other metabolic processes that we've talked so far are also happening. And biosignaling as well connects everything, and it requires regulation and integration and working together as a whole, not just it's the cell itself, but with all the cells together. Imagine how magnificent that is and how humbling in a way it is. Now, to get back to translation, you can see here that I've listed a lot of questions, and normally if we had a discussion in class, I will ask you because I expect that you already know that. So I'm going to go through these questions fairly quickly, answering them since obviously we're doing this via video. So what determines the protein sequence? You already know that the newly synthesized protein, or the sequence of the newly synthesized protein, depends on the nucleotide sequence of the mRNA. Why is this process called translation? I wonder if you asked yourself that question. But if you think about it, the language of DNA, of the nucleic acids in general, DNA or RNA, is made out of 4 letters, A, G, C, T, or A, G, U, C, for RNA. And then that language is translated into a product made out of different letters, or from a different alphabet that has 20 letters. That there are 20 letters in that alphabet, and those are the amino acids. So for that reason, the process is known as translation, because we are transforming one type of information written in one way to another type of information written in a different way. And when you think about it, for any translation to occur, you need to have an interpreter or translator. In this particular case, the molecule that makes this possible is actually tRNA, shown here as the adapter. tRNA is the one that matches the corresponding amino acid with its corresponding nucleotide sequence. Now, you already know that the position of each amino acid is designated by a sequence of 3 nucleotides known as a codon. The question that I wanted to ask you is, why 3? And the answer to that is because we're matching 4-letter alphabet with a 20-letter alphabet. And obviously we cannot have each nucleotide designate the spot of multiple amino acids. It will be very confusing. For the same reason, we also cannot have a sequence of 2 nucleotides, designating the spot for one amino acid. However, a sequence of 3 nucleotides is sufficient enough and gives enough variability to allow for specific localization of a single amino acid. And you already know that each nucleotide designates the spot of one amino acid, but one amino acid may require or may be coded by one or multiple nucleotides. So how many codons are there, and how many of them code for amino acids? There are obviously 64 codons. How do we know that? Well, simple math. We have 4 nucleotides that are grouped in 3. So if you do 4 on the power of 3, you'll get 64. How many of them, are there any of them that do not code for amino acids? There's 3 of the 64 nucleotides that are known as terminators or stop codons. Why the discrepancy between the number of codons and the number of amino acids? Well, simply we have 64 of them, only 20 amino acids, so we're bound to have some discrepancy where some amino acids will be coded by multiple codons. How did scientists find out which codon matches which amino acid? It's a very interesting question. In the very beginning, much of what we take for granted now and we learn about even in high school was not known just 50 years ago. Or maybe not that 50, maybe about 70 years ago. Either way, this was established experimentally. Basically, the investigators first generated mRNAs that are made out of one nucleotide. For example, poly A or poly U or poly C mRNAs. And the resulting products of these mRNAs, for poly U, for example, was a polypeptide made by phenylalanines. For poly C, a polypeptide made out of polyprolines. For poly A polypeptide made out of lysines. For the G, though, this turned out to be a difficult task, because when you make an mRNA made out of only G nucleotides, they form very complex interactions between each other, and they couldn't bind and they couldn't be used by the ribosome. Later on, they used different combinations between J, A, G, C, and U, and that is how scientists were able to determine which codon codes for which amino acid, or they were able to determine the genetic code table. All right, well, the next question we should ask ourselves is, where does translation occur? And I'm sure you already know that this happens in the cytosol. While replication and transcription occurs in the nucleus, translation does occur in the cytosol. Scientists also were able to figure out that tRNAs needed to be bound to the amino acids to activate the amino acids, but at the time, they didn't know how the tRNAs related, or how exactly they relate the amino acids to the mRNAs. Using in vitro approaches, researchers showed that amino acids bound to their corresponding tRNAs bind only to specific codons and nothing else. In this interaction, the tRNA matches the amino acid to the given codon of an mRNA, basically shown in this table, where the phenylalanine tRNA complex will bind only to the UUU trinucleotide, where the lysine tRNA complex would only bind to the poly A, and the proline tRNA would only bind to the poly C. And that's how they were able to ascertain that the amino acyl tRNA complex is necessary, and specifically tRNA is necessary to match the amino acids to its corresponding codon. Now, we already mentioned that we have 64 codons shown here in this table, and that some of them have a special function. We already mentioned the three, the highlighted here in red. These are the stop codons. And one additional one, the AUG or ATG codon, that is the methionine codon or the start codon. Now, for any sequence that has 50 or more nucleotides, and that sequence has a start codon, ATG, and finishes with a stop codon in frame with the start codon, that sequence of 50 or more nucleotides that has a start codon and a stop codon in frame is known as an open reading frame, or ORF, and it codes for a given protein. A good question would be, if you have a nucleotide sequence, would you be able to determine the protein sequence? The answer to that is, I'm sure you already know that, yes, we could tell the protein sequence if we know the reading frame. That is the key part. You can always translate any triplet of nucleotides and can always match it to amino acid, but the only way to really know whether that nucleotide does code actually for that amino acid is if it is in frame with the start codon. The next question is, if you have a protein sequence, would you be able to determine the nucleotide sequence? And the answer is yes, but to an extent. And you probably will not be able to well, most likely you will not be able to determine the exact sequence because the codon polymorphism for each amino acid. In other words, as you can see, alanines, for example, are coded by multiple codons. Again, one codon codes only for one amino acid, but one amino acid can be coded by multiple codons. Okay, obviously, we've talked about the importance of the tRNA as the adapter, matching each amino acids to its corresponding codon. We know that 61 codons code for amino acids. A natural question would be, do we have 61 anticodons that are part of the tRNA structure that help the recognition or the matching of the corresponding amino of the amino acid with its corresponding codon? The answer is no. We don't have 61 anticodons. We have less anticodons, but there is no specific number for all mammalian cells. How the anticodons interact with the codons is explained by the Crick's-Wobble theory. The tRNAs have an additional nucleotide, the I, or the inosine nucleotide, which allows for certain level of flexibility with respect to base pairing. Now we're gonna, well, I don't have time to go into the Wobble theory in this video, but I will continue with that in our next recording. Aloha, welcome back to our discussion. We will continue now with discussing the Wobble theory, which describes how the codon from the mRNA associates with the anticodon from the tRNA. There are four postulates. The key part here is for you to pay very close attention to what is being described and whether we are talking about the codons or the anticodons. That will help you stay focused and not getting confused, because it's very easy to get confused and mix things up. Okay, so the first postulate: the first two bases of a codon always form classical Watson-Crick base pairing with the anticodon. I'm going to use this example right here. The codon obviously in 5' to 3' direction. The anticodon is in the opposite direction, 5' to 3'. What this postulate says is that if you have an A and the first base or the second base of the codon that A will always associate with an U with the anticodon, which is either in the second or the third position of the anticodon. Or if it's a G or a C, it will obviously make a base pairing with a G if it's a C, or C if it's a G. Classical Watson-Crick base pairing. And when we're talking about the first or the second base of the codon. The second postulate, the first base of the anticodon determines how many codons the tRNA can recognize. Which basically means that based on what base will be on the first position of the anticodon, we will have different number of codons being recognized by that anticodon. So for example, if the first base of the anticodon is an A or a C, these anticodons will bind with exactly one codon. If the first base of the anticodon is a G, we will recognize that anticodon will recognize two different codons ending with either C or a U. If the first base of the anticodon is in U, that anticodon will recognize two different codons ending either with an A or a G, provided that the other two are classic Watson-Crick interactions. And if the first base of the anticodon is an I, that anticodon can recognize codons ending either with an A, U, or a C. Now the third postulate, if an amino acid has more than one codon and those have different first or second bases, such codons will require different tRNA. And the last postulate, a minimum of 32 tRNAs are required, or anticodons are required for the 61 codons. Now to our last question, which was basically your assignment for today. If you were given an mRNA or a cDNA sequence, how would you know which sequence is the coding? Now, when you have an mRNA, or more likely cDNA sequence, you start from the beginning searching for the first ATG codon. Let me take a step back, just to remind you the difference between mRNA and cDNA. An mRNA, the sequence of an mRNA is basically the same as the provided sequence of the cDNA. If you remember from 481, cDNA is a copy of the mRNA, but instead of U, it has T nucleotides. So they're the same, because cDNA means copy DNA or complementary DNA. Okay, so when you are given a cDNA sequence or an mRNA sequence, the way you will identify the open reading frame is by simply scanning, starting from the beginning, and start searching for the first ATG codon. So you scan down the sequence, and whenever you hit ATG, that will determine the start of the open reading frame and the frame itself. From this point on, the three nucleotides following the ATG will form the next codon, and then on and on and on until we reach a codon, one of the stop codons. For a nucleotide triplet to be called a codon or a stop codon, it needs to be in frame with ATG. They cannot be overlapping. So they're sequential triplets of bases. Okay, so that's how you determine or can figure out or determine an open reading frame. Okay, normally I would say that this assignment that I've given you, where I provided you with a sequence, you usually struggle with it a little bit or more, and normally I give you the points for that assignment. no matter what, whether you're right or wrong, I'll just give you the points. However, next time you will have a very similar assignment, slightly changed, but for that one, I will watch, or Gracie will watch for the correct answer. Okay, so I'm going to move on. All right, so when we talk about translation, it is natural that we talk about the different stages in translation. Now, we begin translation with amino acid activation, where the tRNA will be attached to its corresponding amino acid, forming amino-acyl tRNA complex, and that reaction is synthesized by the amino-acyl tRNA synthetase or synthase. Why is this necessary? Well, there are two reasons for that. First, you want to match the tRNA to its amino acid, because the tRNA will deliver the amino acid to its corresponding place. And second, to activate the amino acid. That amino acid will be involved in the peptide bond formation. You must have the carboxyl group activated if there will be an attack by the amino group of the following amino acid. We'll talk about that later. The carboxyl group must be activated, and attaching it to the tRNA represents that activation or activates the amino acid. The next step is initiation, during which the translation complex is formed with the proper positioning of the start codon. The following stage is elongation, where we extend the polypeptide chain, followed by termination, designated by the stop codon. After the peptide is synthesized, things are not done completely. We have folding and post-translational processing, which we're going to discuss later. Now, obviously, the workhorses of this process are the ribosomes. The ribosomes are made out of rRNA and proteins. You can see represented the structure shown here. Everything in gray is rRNA. Everything in blue, the ribbons, are proteins. You can see that the majority of the ribosomes are actually rRNAs. So in essence, these are ribosomal enzymes or ribozymes. Everything in the active site is actually, the active site involves only ribosomal RNAs and no proteins. The tRNAs shown here in this inverted L shape are single polypeptide chains that are made out of, well, usually anywhere between 73 and 93 nucleotides, and they're folded around each other. Okay, now let's talk about the amino acid activation. The activation process occurs in the cytosol. The interesting thing here is there is one amino-acyl tRNA synthase per amino acid. In other words, we have 20 of them. If more than one tRNAs are allowed per amino acid, the same enzyme will synthesize them, or will match them together. So one enzyme per one amino acid. There are two types of amino-acyl tRNA synthetases based on where do they initially attach the amino acid. We'll talk about it a little bit later. The first step of the amino acid activation is amino acid adenylation, shown right here with the help of ATP. Now we have an amino acid that has been adenylated. That, in a way, is the activation. Then the amino-acyl tRNA synthase or synthetase, based on whether it's class 1 or class 2, will add the amino acid either to the second prime carbon of the ribose of the first nucleotide of the tRNA, or to the 3 prime carbon hydroxyl group. Class 1 adds to the second prime, class 2 adds to the third prime. Regardless whether we initially add the amino acid to the second or the third, they all will end up being added to the hydroxyl group bound to the third prime carbon. It's just that for class 1 it takes 2 reactions, where for class 2 it's just one single reaction. Here I'm asking you to realize the purpose of these steps. First we have an activation with the help of ATP, and then that activation will be carried by that amino acid being added now to the first nucleotide of the tRNA. In essence, that carbonyl group or carboxyl group is now being activated for the follow-up reactions. This process also involves proofreading. The errors are 1 in 10,000. The proofreading is multi-layered. Either we can proofread the amino acid enzyme matching. If the wrong amino acid is added, if it doesn't fit the active site, is being expelled. If for some reason the wrong amino acid is part of the active site and we form the wrong amino acyl AMP or adenylated amino acid, it won't fit in the active site and will be hydrolyzed and the wrong amino acid expelled. If the wrong amino acid gets activated and somehow gets added to the wrong tRNA, it still won't fit in the active site, and then it will be hydrolyzed and then expelled. After the amino acyl tRNA is generated, then now this complex is ready to be matched with the proper codon. Now, the recognition of the amino acyl tRNA synthase of the corresponding tRNA for the proper amino acid is referred to as the second genetic code. In other words, it is very important for the amino acyl tRNA synthetase to recognize the proper tRNA and match it with its corresponding amino acid. That ability or that recognition is referred to as the second genetic code. Now, for the remaining time, let's look at the tRNAs. This is the classic two-dimensional representation of tRNAs with the amino acid arm. That's where the amino acid gets added. These two arms, D and the T-side arm, are responsible for the folding and the recognition of the tRNA by the ribosome. And this arm is the anticodon arm. That's the arm which is used to match the anticodon with the proper codon. Many of these nucleotides that are part of the tRNA are recognized by the amino acyl tRNA synthetase. Some synthetase requires complex or multiple nucleotides to recognize the corresponding tRNA. Some use just one. For example, for the alanine tRNA amino acyl synthetase, to recognize its corresponding tRNA, we only need to have this GU base pair, which is required for recognition. I'm running out of time. I'm going to record a third video so that I can explain this slide a little bit better. So I'm going to end it here right now, but I'm going to finish later on just with a few more minutes in the third video. All right, thank you. Aloha. Welcome to our third and last part of the Wednesday lecture. I decided to step back and start again from this part of the presentation. Not because I didn't explain everything, but so that I can connect it with the following slide. Okay, so like I said, translation begins with amino acid activation. The amino acid activation occurs in the cytosol. Something that we need to remember is that for each amino acid that will be paired or connected with a tRNA, there is a unique and separate amino acyl tRNA synthase or synthetase enzyme. Both pronunciations are correct. So we have one enzyme per amino acid. If that amino acid requires two or more different tRNAs because of multiple codons, for example, it's still going to be that enzyme, one enzyme. In other words, the enzyme is specific for the amino acid. Okay, we have two types of aminoacyl tRNA synthetases, class 1 and class 2. They differ based on where do they attach the amino acid, either on the second prime hydroxyl group or the third prime hydroxyl group. The first step in the amino acid activation begins with the adenylation of the amino acid. When the amino acid is adenylated, it is now activated. When the amino acid is activated, it then either gets added by class 1 aminoacyl tRNA synthetase to the second prime hydroxyl group or to the third prime hydroxyl group, which is done by the class 2 aminoacyl tRNA synthetase. No matter which class the enzyme is, all amino acids will end up being added to the third prime carbon hydroxyl group of the first base of the tRNA. Class 1 uses 2 steps, class 2 uses 1 step, but they all get added to the third prime hydroxyl group. Now this process has proofreading as well. The errors are 1 in 10,000. The proofreading happens at the multiple level. First level is the recognition of the amino acid by the enzyme. If the amino acid doesn't fit, the enzyme expels it. The second level is if a wrong amino acid binds to the active site, but then it gets activated by adenylation, the aminoacyl AMP that is formed doesn't really fit very well in the binding site, and then it gets hydrolyzed and the wrong amino acid expelled. The third level is if we have the wrong amino acid added to the tRNA. If that happens, the enzyme has the ability to recognize that the wrong complex has been made, and when it recognizes that, it severs or hydrolyzes the aminoacyl tRNA complex, and the wrong either tRNA or amino acids are released or expelled. Now, obviously, for this to happen, there's a lot of pressure on the enzyme. The enzyme on one side has to recognize the proper amino acid, but on the other side, it has to recognize the correct and corresponding tRNA. That recognition by the enzyme of the correct tRNA matching the bound amino acid is known as the second genetic code. It is very important for the enzyme to recognize the correct tRNA matching the amino acid. On this slide now, I'm showing you the two-dimensional structure of the tRNAs. Here you can see the crystal structure, the ribbon structure. Actually, I shouldn't say the ribbon, it's the stick model of the tRNA. You can see the inverted L shape of the tRNA. This representation is just for simplicity and just so that we can see everything a little bit better. We have the amino acid arm, which binds with the amino acid. We have the D arm and the T side C arm and also we have the extra arm that not all tRNAs have. But these arms, they're called this way based on the type of residues they have, and you can see the different residues right here. But these two arms have a role in the tRNA folding, and the T side C arm also interacts with the ribosomal subunit. The anticodon arm, the name explains itself, is the arm that contains the anticodon. It's used to bind with the corresponding codon on the mRNA. Okay, you may ask on this image, why do we have these different dots, different colored dots? Well, these are recognition dots. Some of these are used for recognition, some of them are not. Those that are colored in blue are the same in all tRNAs and cannot be used to discriminate one tRNA from another. On the other hand, those that are colored in orange or in green, right here, are used either by one or more amino acyl tRNA synthetases. Remember, the synthetase need to be able to recognize the correct tRNA that will match its corresponding amino acid. Okay, multiple nucleotides can be used for recognition of a tRNA by the synthase. In some cases, recognition can be very simple, and in some cases, more complex. The recognition of the tRNA for carrying alanine, for example, is a simple GU base pairing in the amino acid arm of the tRNA, right here. Now, if we compare how well would its natural tRNA be recognized by the alanine tRNA alanine tRNA synthetase, how well will this tRNA be recognized by the enzyme, or this truncated version will be recognized? When we compare them, both of those versions get equally well bound to alanine, which basically tells you that the recognition for the tRNA correspondent to alanine is all connected or all determined by this GU base pair right here in the amino acid arm. You can cut this entire part off from the tRNA and the product will still bind to alanine and deliver it to the corresponding codon, if of course we have an anticodon. Well, just for the sake of the conversation, this truncated version will not effectively deliver alanine to the ribosome and obviously be involved in translation. It won't fit. It doesn't have all the other recognition sites. All we're studying here is what are the recognition signs, or what are the recognition parameters for the amino acyl tRNA synthetase to be able to match the correct tRNA with its corresponding amino acid.